Hello, everyone, and welcome to our webinar, Using COSMIC to Predict, Identify, and Avoid Mutational Consequences of Cancer Therapies During Early Drug Development and in Patients. I'm Caitlin McCann-Jankovitz, and I'll be your moderator for today's interactive event, brought to you by QIAGEN Digital Insights, or QDI. This webinar will be available on demand following the live presentation. We'll alert you via email when the recording is available for replay. All right. I'd like to now welcome our speakers, Dr. Kyle Nilsson of QIAGEN Digital Insights and Dr. Steve Jupe of COSMIC Wellcome Sanger Institute. Dr. Kyle Nilsson is a sequencing-focused molecular biologist and a field software trainer at QIAGEN Digital Insights. He earned his PhD in molecular and cellular biology from the University of Iowa, where he studied the regulation of RNA polymerase II transcription and co-transcriptional mRNA processing. Dr. Nilsson then completed his postdoctoral training at Penn State and Cornell University, focusing exclusively on NGS method development to study chromatin. Dr. Steve Jupe joined the COSMIC team at Wellcome Sanger Institute as the principal curator for actionability in 2018. His extensive career in biology and bioinformatics includes 7 years as a postdoctoral molecular biologist, 13 years in the bioinformatics group at GlaxoSmithKline, and 9 years curating biological pathways for the Reactome project at EMBL EBI. And last, before we get started, our legal disclaimers. QIAGEN products shown here are intended for molecular biology applications. These products are not intended for the diagnosis, prevention, or treatment of a disease. For up-to-date licensing information and product-specific disclaimers, please visit www.qiagen.com. A license for COSMIC is required for commercial use of data obtained from either the website or downloaded files, which ensures the continued growth and maintenance of the resource for academics and industry alike. For additional details regarding the commercial use of COSMIC, please visit the URL that you see here or contact bioinformatics sales at QIAGEN.com for complete terms and conditions. And with that, Dr. Nilsson, I will pass it over to you to get us started.
Awesome. Thank you so much for the introduction, everyone. Thank you for taking the time to attend today's webinar. I'm really looking forward to showing you some of the really interesting ways that you can use variant data encoded within COSMIC to really tackle some interesting problems that you might encounter in your own research, your own drug development, or also just challenges in staying current for clinical diagnosis. Because there are so many different ways that you can take advantage of COSMIC, I like reminding people that QIAGEN products come with assistance and help. And the COSMIC team is also very interested in learning how you're using their data and how to help you get the most out of that data. So feel free to reach out to either of us. We have our contact information currently on screen, and we will be sure to share this information as well. So today, we wanted to discuss some of the interesting ways that you can encounter mutational evidence of, let's say, deleterious effects. So as an example, you might be developing a CRISPR therapeutic, and with that, you would be screening, you would have a large number of potential targets, and with those targets, you would also have predicted off-target effects. Something that you can use COSMIC for, and that I'll show in the first use case, is you can dig into the genome location information and look either on the website or systematically using the downloaded files to prioritize and interpret many of these variants to see which particular modes might be dead ends, which ones might be better suited for your time. In addition, COSMIC has a wealth of incredibly valuable summarization and curation put into it. What I mean by that is that in addition to being able to download the various COSMIC information that you would expect, the annotations that you want, you also have access to a lot of effort put in by scientists like Steve, where they're focusing on the broader biochemical context that's leading to mutations to become drivers of cancer. You can find that kind of information in the Cancer Mutation Census project. And we also have the Mutation Actionability and Precision Oncology set of data. And that project really focuses on linking individual variants to their diseases, as well as to potential clinical treatments. All of this information can be super valuable, both for discovery scientists who are looking for projects, looking for interesting ways in which they can link their variants of interest to existing therapies, and also in the clinical space where you really are looking for the cutting edge of how you can help your patient. Speaking of the cutting edge, the final thing that I want to discuss today is mutational signatures. And this is where we are dealing with growing evidence of large, multifaceted aspects of variants occurring in individual cancers. So for that data, COSMIC, again, has been at the forefront of annotating and curating this information. And its goal is to be the trusted, carefully considered source of these mutational signatures. So although there are not many signatures that are currently in clinical use, that is going to change over time. And I'd like to show you how you can use the mutational signature evidence in COSMIC to stay appraised of, let's say, potential consequences due to chemotherapies. So there are a number of signatures that have been linked to individual chemotherapies or other treatments. And those are things that it can be very valuable to keep an eye on so that you don't accidentally give an inappropriate treatment. So overall, the agenda that we will focus on today is going to be those 3 use cases. But in addition, I also want to give just a brief introduction at how QIAGEN, as well as our partners with COSMIC and other data providers, all of the cool curated content we have, and how we really insist on quality of data. So with that, I'll just go ahead and jump into that super brief introduction to show how all of these resources can potentially link together. So first, why do we insist on expert curation of public data? Why do we rely so heavily on scientists in order to manually validate many of these findings when we're in the era of machine learning and natural language models? Simply put, scientists make mistakes. I know that from my own experience, I have certainly filled out metadata tables uploading to Geo where I used Excel to drag and drop my cell lines, and all of a sudden I have K562, K563, K564. A scientist can see that error and fix it immediately. They know exactly what happened. They know exactly what was intended in the original metadata table. But if you're relying entirely on language learning ingestion, then you run the risk of having false information, or worse, you might miss complete sample swapping or other forms of really deleterious data. So because of that, we really like being proud of the fact that we are a leading provider of expert-curated knowledge. And we like to think of ourselves as your data partner in how we are able to provide a variety of these curated data sources. For example, we have curated research findings. These are the data that form nodes and relationships showing how individual genes might be connected to each other or to diseases, drugs, chemicals, etc. And that information is something we curate in our biomedical knowledge base, in this case, our human-derived biomedical knowledge base. And that data ultimately powers ingenuity pathway analysis, something that many of you are familiar with as a tool to both analyze your own comparisons as well as explore this public data. In addition, we curate carefully omics data sets. So our omics lands contain hundreds of thousands of samples where we have taken the time to reprocess the data from scratch and ensure the metadata is using controlled vocabulary so it's cross-searchable. The subject of today's talk, though, is the curated gene variants, the mutations that are ultimately found in both cancer as well as rare disease patient samples. So today we're going to be talking about COSMIC, but we also have our own somatic mutation database, which focuses pretty much exclusively on clinical samples and frequencies, as well as our other data partner with HGMD Professional, where we focus on those germline mutations. But something that is really important that I like mentioning is that COSMIC is so trusted as the leading source of somatic mutations in terms of its breadth and comprehensiveness. We actually use it to power IPA as one of our data sources. So again, today we're going to focus on COSMIC, which we've recently updated. So version 99 came out at the end of November, and we're actually coming up on the version 100 release. So definitely pay attention for that coming out. But with this current release, we have over 24 million genomic variants. We have tons of coding and non-coding information, millions of samples, and a large amount of side projects that ultimately form the Cancer Consortium data that you would expect from the gene census as well as the mutation census. And then also the actionability information that is really important for linking cancer to disease. So that was hopefully a brief summary of just the kind of data that we like curating here at QIAGEN and the kind of data I love playing with as a scientist who works here. So with that, I'm going to go ahead and jump into the first use case that I'd like to present today, which is using COSMIC to potentially identify and prioritize gene editing targets. So essentially, how can you use COSMIC to look up genomic locations? How can you use this to ultimately integrate that data into your own pipelines and development? So here, I just have an overview of the slide where I'm just describing kind of the whole story, where you can use both COSMIC's website as well as the genome browser in order to manually scan regions of the genome for potential mutations and then pull up information about them. Something that is extremely important, though, is linking that information to priority information. Essentially, how are these variants classified, especially in terms of whether or not they are drivers of cancer? What is known about their pathogenicity? And ultimately, in order to get to that information, you do have to rely on the ability to download the data. So the COSMIC Cancer Mutation Census information can help you look beyond just what you can see in the genome browser and focus instead on pulling out individual variants in a more systematic way. Or if you happen to be looking specifically for pathogenic variants, a simple search of the data files can help you identify what those are. And then again, you can search within those genomic regions to pull out particular areas. In this case, variants around this gene, which have documented cancer activity and evidence of functional impact. So overall, the summary of what I want to show is that it's possible to download and integrate COSMIC data into your analysis pipelines. So with that, I want to go ahead and just share my screen briefly so I can show what I mean by some of these examples. So here on screen, I currently have my COSMIC web browser. And this is where you have access to a large number of different resources, regardless of whether you're an academic or a commercial user. But once you're able to log in, you can always search for your particular gene of interest or your region of interest. Something that I also like pointing out is that there is the ability to download data once you've registered. So here I'm showing under data, we have this downloads page. And then once I click here, this is where you can see a variety of different file types, where here in version 99, we have all of the variety of data sources, including hallmarks of cancer. We have non-coding variants. We also have some of the really important things like the Cancer Mutation Census, which in this case, this Cancer Mutation Census data is what I'm going to be using to power some of today's queries. That said, this is just one of the ways that you can access this data. And this is how academics would probably register in order to download. But if you happen to be a commercial user, you can also log in through your QIAGEN Digital Insights portal. And this is where, if I log in here, it'll give me a prompt to log in. And then once I've done so, this is going to give me the ability to then look at all of the various data resources that I've subscribed to. And here, this is where we have access to the COSMIC downloads. So after checking the version that I'm interested in using, in this case, I want to use the public release of version 99, this is where I'm able to find details about our actionability release notes, details for the Cancer Mutation Census, the actionability data and CMC data itself. And then scrolling further down, you can see that all of the TSV flat files are available. So that way you can download and integrate them into your own pipelines. So all of these details are detailed very extensively, both on the COSMIC webpage and also the journal articles where the COSMIC team has taken the time to really go over what is contained within this resource. So first, let's say that I am interested in looking around a particular gene region. So a screen for genes involved in T-cell receptor signaling happened to come up with TNF-AIP3 as a potential regulator for T-cell activation. And this actually was a lentiviral transduction and Cas9 CRISPR screen in human CD8 T cells. So if I'm a researcher, I might want to start by searching for this gene. And then when I do so, this is where I can go ahead and pull up the various gene details and pull up as well the genomic browser information so I can see exactly where in the gene and surrounding regions there are known mutations. And as you can see, COSMIC comprehensively reports variants whenever they are encountered in the literature. If it's something that can be curated and verified, it's something that is likely to be included. And not all of these variants are of particular consequence. So perhaps instead, you might be interested in drilling down to a very specific region. In the cancer genome browser, you can either type in that region here, at which point this is going to pull up just where some of the mutation sites are located around the transcription start site. Or something that I prefer doing is taking advantage of scripting. So here I happen to have created just a really simple shell script which parses the curated coding and non-coding variants. And once I have looked at these, I can see that when glancing around this region of the genome, I'm able to identify what variants are represented, and also if there are any additional cancer mutation census calls. So for this particular region, this is where we have variants within COSMIC, but none of these have been identified yet as being potentially causative. So what if instead I wanted to search out some of those regions that I might want to avoid? So here again, I'm just looking at this gene, but instead, what if I want to perform a simple search? So in this case, that simple search that I might be looking for could be, show me all pathogenic variants around this gene. So all I'm doing at this point is doing a simple search for TNF AIP3 and whether or not the last column ends with the word or contains the word, or excuse me, whether or not the word pathogenic is contained later in the line. And this is looking at that cosmic CMC export file. And so here, if I go ahead and run this, this is where I can see that there are, in fact, several regions that do have evidence of pathogenicity, some of which are likely benign, some of which don't have fully provided information. But then there is also this one in particular where I can see information about how this particular gene variant might be linked to, in this case, potentially an autoinflammatory disease. And overall, you could look for these regions as well in the genome browser once you've identified them in these mutational files. And here you can see this is quite extensively mutated for our region. So this is an area that you would probably want to avoid if performing any kind of gene editing. Or, of course, you can search more specifically using the flat files. In this case, I happen to have searched around this particular window for this region, and not only was I able to find the annotated mutations, but some of these also had additional Cancer Mutation Census information provided. So in summary, what I hope that I've shown is that it is possible to integrate information from the Cosmic website, but also the really valuable curations present in the Cancer Mutation Census and other resources that you can only access by downloading them. So if you're an academic researcher, please create an account, sign up. If you're a commercial user, I hope you can really see the value in using this to jumpstart your research. So with that, I think it's about time to pause for our first question break.
Yes. Thank you, Dr. Nilsson. To the audience, if you have any questions about what was presented in this segment, please submit them now if you haven't already. That said, we do have some questions, so we'll go ahead and get started with what we have so far. First question. Are there tools for uploading a VCF file and getting automated variant prioritization and annotation as output?
Yes, there definitely are. So one of the tools that we would have available for doing that, let's say you were more interested in a scripting approach. In addition to the scripting options that I have shown, there is also availability of public tools such as Anavar. So Anavar is a really powerful Perl program which you can use to potentially link all of the cosmic information together. And importantly, as you can see, as of last month, it has been recently updated to handle the new cosmic data file formats. And Anavar is something that is accessible as well on the commercial webpage. If, on the other hand, you're looking for more of a turnkey solution, that's where I would actually recommend looking at something like QCI Interpret. And that is a pipeline that we provide where it allows you to upload your own VCF file, whether it's a VCF file containing somatic or hereditary information, and you can use that to pull and prioritize all sorts of information from these variants, whether it's from Cosmic or the greater QIAGEN knowledge bases.
All right. Next question. What is the difference between the mutant calls in cmc.tar and cosmic mutant export.tsv.gz files?
That is a very specific question. To answer that one, this is where I would probably delve into the files themselves. But something that I can always do to double check is there are the release notes and the README, which does contain in a very detailed format exactly what columns are present in every single file. So this would be a resource that I recommend looking for the answer. Although I don't have the answer on the spot, this is where I would look in order to, say, respond by email.
All right. And last question for right now. How does Cosmic choose papers for curation, and what types of data do they extract?
That's a really good question. And for that, I think one of the best places to look actually is the Cosmic publication. So here, if I just scroll down to this second figure, this is where I'm able to pull up an example. Actually, I'm going to not wait for the figure to load. Here, essentially, we're looking at a variety of sources from PubMed and other publications. And my understanding is the Cosmic team is scanning on a weekly basis for any of the various keywords that are associated with somatic mutation. And once it has compiled those, their team is manually checking those to see, is that in fact a somatic mutation, and can I go ahead and import that information? And when the Cosmic team imports that, these are the features that they happen to be looking for. Essentially anything related to the patient, any information about the tumor, any information about the sample, the gene itself, and also what exactly is the mutation? Is it a heterozygous? Is it homozygous? What are the coordinates? Ultimately, not every piece of data is always available, but the items shown here in red are things that are mandatory. So Cosmic will always pull information on the gene, the mutation syntax, the primary site, and the histology data.
All right. Dr. Jupe, did you have anything to add to that?
No, very comprehensive answers. Thank you, Carl.
Fantastic. Okay, and we did get one more question in that time, and I think we have adequate time to answer it. So one more. Is there any plan to classify the tiers according to standard guidelines such as AMP?
That's a great question. So that's something that QCI Interpret already does. So if that is something that you require, I would look into QCI Interpret or QCI Interpret Translational for those who are more involved in discovery research. However, Steve, you might have additional information about actionability.
Yeah, so for actionability, we obviously looked at the existing standards for classifying actionability into tiers. And you're probably already aware that there are many systems used around the world, and they don't necessarily agree with one another. So we decided to come up with a classification system that had the benefit of being something that removed any subjectivity whatsoever. It's based entirely on the information you can find from the trial data. It doesn't rely on having access to clinical experts to assign those tiers, those categories. And it's not intended to replace any of them either. That's another key thing to say. And I'll talk about the tiering, the ranking system used in actionability later on.
All right. Thank you, Dr. Jupe. And speaking of Cosmic's curation guidelines, this is actually the perfect time to hand it over to you since you are the principal curator for actionability. So feel free to take it away.
Okay. There we go. Excellent. Right. So this image is intended to represent the mutations that you might find in a typical sample. So the blue square is all of the mutations that you might have identified in your sample. And then the tiny yellow area is the mutations within that sample that might well be drivers, that have caused the tumor in the first place. And then the really tiny orange square is the subset of those that are likely to be the actionable mutations. So the variants that give you the opportunity to pick some kind of a therapy that you can use to treat the patient that the tumor sample came from. Moving on to the next slide. So the mutations the mutation itself is useful information, but you need to see it in the context of the disease. So here's an example of mutations within the KIT gene. On the left, we have mutations in mast cell neoplasm, and you'll see that 72% of the samples that we have for this disease have a KIT mutation. And a very common mutation within that disease is this D816V mutation. If you contrast that with what you can see on the right-hand side in gastrointestinal stroma tumor samples, where 55% of those samples have a mutation in the KIT gene, but it's in a completely different place. The most common mutation in that disease is in a completely different place, unlikely to be driving the disease in a very different way. So it's really important to have access to deep information, many samples obtained from for these diseases, so you can make this kind of comparison. And COSMIC has 30 million coding mutations in 1,400 different cancer types, which will hopefully help you to characterize the genetic events that will actually characterize what's going on and driving the disease. Okay, so when you're faced with 30 million mutations, how do you decide which ones actually matter? Well, if you have enough depth of information, you have access to that data, you can, and you have some expertise in doing this, you can do that for well-characterized genes. But what do you do if you have a gene that has no well-understood mechanism? And what do you do if you've got 50 genes or maybe 2,000 genes that you want to be looking at? Then you have more of a problem. So how do you then identify the mutations that actually matter? Well, one way you could try to do that is to look for variants that have been described in the literature as being highly significant for a particular disease. Or you can look at the recurrence of mutations in cancer samples as a reflection of if you see a variant that's happening very frequently in a particular cancer subtype, that's reflects that it might be positively selected within that cancer type. Or you can use various algorithms for identifying significance of variants. But those algorithms, there are some problems. The main one is that they're frequently black box. They're based on correlations, and you can never be quite sure how accurate they are. So, and there's a caution about whether recurrence is actually an ideal indicator. So sometimes recurrence may not be a good indication that a mutation is particularly significant. So here's an example using views from within COSMIC. This is the ALPK1 gene. And you can see within ALPK1, there are these very strong peaks of variants in these positions that you might look at and think, well, those peaks, they seem to be occurring pretty frequently, so there must be something significant going on. This gene is a serine threonine protein kinase. It's involved in the detection of pathogen-associated molecular pattern metabolites, or PAMPs. So it's part of the immune response system, which is, of course, critical for eliminating pathogens and also for the engagement of adaptive immunity. So those peaks of highly recurrent missense mutations are quite typical for oncogenes. However, yes, so the problem here, what's actually happening, is that despite the best practice bioinformatics processing that's used, and also the benefit of expert curation for everything that goes into COSMIC, somatic variant datasets can still be contaminated by germline variants. So all of these highly recurrent variants that were identified that you can see within the ALK1 gene have actually also been reported as germline variants in normal populations. And the reason why you might see these polymorphisms so commonly is because this is a gene involved in innate immunity. There might actually be some advantage to that. So on the right-hand side, we have an image from the CMC data. So in the CMC data, you've got additional parameters here that might help you to recognize the fact that what you're actually looking at here is a peak caused by a germline polymorphism rather than a variant that's actually somatic. Okay. So CMC, the Cancer Mutation Census, is the cosmic resource to help you reduce this uncertainty. It takes into account the germline variant frequency and uses a number of different measures, which are the boxes on the right-hand side, to help to try and identify which somatic variants are the ones that are actually pathogenic as opposed to benign. Oops. And so within CMC, based on a number of clearly stated criteria, we prioritize the mutations that have the highest potential to be driving cancer into tiers. And there are 3 tiers. They're represented by by colors, by dark red and orangey red, and then pale green, and then everything else is gray when you're looking on the CMC web pages. Okay, so this is a view from within CMC, and basically we have a view of a whole gene on the left side, and then zoomed in to a more precise view on the right-hand side, where we've zoomed into an interesting region. So you can see on the left-hand side there, on the left side of the bar towards the top of the screen, there are some red lines, and then we've zoomed into that region on the right-hand side to get us to inspect the mutations at that location, and also see the rows beneath it give you an idea of the measures and the strength of the measures that have been used to assign those tiers. Okay. You can also look at an individual codon. If you select a single codon, you get a view that looks like this. With this clock face view, one of the interesting things here is that we have multiple codons at the same position, different amino acids at the same position, all have their own set of scores. And the idea of this clock face is that the length of the hands is an indication of the strength of the individual pieces of evidence that contribute to that overall tiering score. Okay. Yes, and so for every individual mutation, there are a number of parameters that are basically collected to give you an overall, to give the overall tiering score. And you can examine each of these in great detail. Okay. So that's CMC. This is now talking about COSMIC's actionability data. So the full name of that is COSMIC Actionability for Precision Oncology. So the aim of this resource is to represent the availability of drugs targeting somatic mutations at all stages of drug development, from the through the clinical development phases up to and including regulatory approval and availability on the market. Okay, so so some of the questions you might want to answer with a resource like this would include, fairly obviously, I have a patient who has a tumor. They have a particular variant present in that tumor. Are there any drugs that I can use to treat such a patient, which have actually been approved by the regulatory authorities? You might also want to know, are there any late-stage trials with results that are encouraging, that means that this is a drug that's quite likely to come onto the market in the near future? You'd probably also like to know if there are any drugs that are undergoing clinical testing at the earlier stages, the things that are coming up through the development process. And you might want to know whether there are trials that are recruiting patients that perhaps you could register your patients to take part in a clinical trial. So actionability could help you answer all of those questions by recording details of ongoing clinical trials. And the key things that get recorded are the disease that the patients have within the trial, the variants which they're either required to have or will be looked at as part of the results when they come out, and also, of course, the treatment that's being given to the patients when they take part in the trial. So we record this information, and we record the mutations in as great a level of detail as we possibly can, depending on the information that's actually provided in the trial details. So obviously we would like, ideally, to identify point mutations. I should say all of the mutations in actionability are defined at the amino acid level because it's exceedingly rare to get cDNA level representation of variants in a clinical trial situation. But if a point mutation is actually required for the patient to be included in the trial, then we will record that. Sometimes the trial will only say mutations in a specific gene, and then we can record then the sort of less precise level of granularity is being used to recruit patients into that trial. And you'll see that there are we have a number of categories for the level of precision. The treatments can be any type of treatment. They can be small molecules, antibodies, chemotherapy, radiotherapy, and of course combinations of those, which is increasingly important in clinical trials. And the details we record are all the things that you'd expect: the phase, the status of the trial, primary completion dates, and of course, and the primary objective, the thing that's going to be used as a measure of success of the trial. The results, when they become available, will include anything that anything that's been measured effectively, and the source of where the results came from, and which is important because clinical trials often come out in more than one form. The results become available as interim results. There may be several publications. Sometimes the results that you see on the the drug label might be different to the results in the publication. So you need to say which set of results you're referring to. And we also record something that I call a progression remark. And it's used to record unusual outcome measures, whether there are any serious adverse events noted. For trials that have been terminated prematurely, we'd actually spend trying to find out why the trial was terminated, which is potentially useful for drug repurposing. And we also look at things like the correlation of the variant with the efficacy, if that information is available as well. So where does the actionability data actually come from in COSMIC? A lot of it has come from clinical trial database records. So clinicaltrials.gov is probably the greatest contributor to that. And that's a great resource. It's freely available. But there are some problems. The main one is that it's often incomplete or somewhat inaccurate when compared to the information that you can find elsewhere. It's unfortunately the case that filling in clinical trials databases is something of a gentleman's agreement. So very often, particularly results of trials won't become available in those databases until a considerable time after they've been published, and sometimes, often, in fact, not at all. So we also look in the literature to try and identify the existence of trials and the results of trials. So not all trials are recorded in the trials databases. You have to look in the literature as well. And not all trial publications are available in PubMed, which is something else that people don't always appreciate. Very often the results will be in more obscure journals, or they might be only available in conference abstracts, or possibly even on the company web pages. So we look in all of those places to try and identify results. And the outcome of that is that COSMIC's literature curation identifies something more than 50% more results for trials that should have results than you would find if you just look in clinical trial databases. I already said we do investigate why trials have been withdrawn or terminated, and if we can find that information out, then we'll provide it. We also include the details of all the trials that were submitted to the FDA, to the regulatory authorities, when a trial is when that information is submitted for a trial to be approved and made available on the market. Okay, so the principal units of actionability then are the mutations themselves. And those mutations are, when it's possible to do so, they're cross-referenced to core COSMIC records using COSB and COSF identifiers. The disease annotations we use COSMIC's internal cancer classification scheme, which is represented in the download files by a COS-O ID. Drugs, in fact, treatments, associated with trials, where possible, we provide Campbell IDs as an external reference for those. But in fact, some of the drugs are so new that they're not actually available in Campbell. When Campbell catches up, we add the IDs. So the sort of the key triple, if you like, of actionability data is the combination of the drug, the mutation, and the disease. So when we've collected all of the trial information, we try and we have a ranking system as a means of suggesting how far progressed are the drugs which are available to treat that particular mutation and disease. So level 1 in our ranking system represents mutation disease combinations that have an approved marketed drug. Level 2 is used for the situation where a drug has been through it's in clinical trials, the results have come out. The results have a statistical measure of some kind, so you can assess a probability score. Where a trial has basically got a promising result, for lack of a better way of explaining it, and those trials in the late stage with promising results are very likely to go to the next stage, which is to be submitted to the regulatory authorities. So these are the drugs that are going to be coming onto the market next. Level 3 is basically anything that's in an ongoing clinical situation with or without results, or for trials where the results have come out and they didn't meet the primary outcome measures. And then level 4 is for variants that we've identified as individual case studies. So we add in case studies largely to expand the coverage of the number of variants within a gene. And this is my last slide, which is actionability statistics. So we had our 11th actionability release last month, and we've now covered fully curated 147 genes and included to some extent 500 and something genes. Fully curated means that we've included all of the trial information that we can identify either in clinical trial databases or in publications, and that we will keep that information updated in the future as well. So we'll go back, we'll look for the results, we'll look for any new trials, and we'll do that for every actionability release, and that happens on a quarterly basis. Okay, thank you. As I said, that was my last slide, so I'll hand back to Kyle now.
Thank you, Steve, for the very, very detailed information about all of the curation that goes into COSMIC, and also for showing the Cancer Mutation Census information in detail as available on the COSMIC website if you register. So for the very last portion of the talk, I want to briefly just mention potential uses both now and in the future for COSMIC mutational signatures. And with that, I have to start by explaining what are mutational signatures. So essentially, when dealing with these mutational signatures, this is where you're dealing with the accumulated mutations, double-strand substitutions, copy number variations, breaks, other kinds of issues that have grown over time and throughout the patient's life. And some of these will be just simply environmental factors or genetics. Others will be because of acquiring something deleterious at some point in their lifetime. And something that many researchers, including COSMIC in partnership with many of these other consortia, so the Alexandrov lab that's linked below, the Pillay lab, also these other resources, essentially they've developed tools to scan many tens of thousands of cancer patients in order to identify these potential signatures. And with that, this is where we can show, potentially speaking, what the signatures are. And ultimately, you have to deconvolute those signatures that you get over time. So for example, currently on screen, let's say that there is a signature that they might have acquired early in the lifetime, where there was some sort of APOBEC delaminase activity. Maybe there was also tobacco smoking in the patient's history, and then at some point in time, a clock-like signature arose. All of these will start at different times, and all of these will thus be present in different parts of the tumor, different parts of the patient, and also to different degrees, depending on that where they started and how long they've had to spread. And so ultimately, this is going to lead to a spectrum of information across the cancer genome. Something that many people will ask is whether or not these mutational signatures are entirely just useful for research purposes, or if they have clinical utility. And something that the Pillay and Alexandrov labs have found is not only can you use this in order to, say, potentially find causative aspects to various cancers, it's also possible to find situations where secondary cancers might be caused by a specific treatment regimen. So, for example, treatment with platinum chemotherapy, this one drug that I can't pronounce today for some odd reason. Many of these compounds have been found to also have their own mutational profiles associated with them. So one in particular, PARP inhibition, has also currently been used. So this is where we can look at homologous recombination deficiency signatures as they potentially arise due to that PARP inhibition. So overall, how do you use the website to evaluate these things? It turns out that the tool that the Alexandrov lab developed, they already use a number of the existing signatures as some of the built-in standards. So from a discovery standpoint, if you're using SIG Profiler in order to assess your samples, you can either take advantage of the extensive standardization that is present in COSMIC, or you can instead build your own standards and specify them, or choose to deliberately exclude certain items. And all of that can be found in the SIG Profiler documentation. If you're a clinical researcher, finding the best method to do this is your challenge, and I wish you the best of luck. But that said, what if you want to instead just keep current on the evidence that might be present for a given signature? So something that is extremely handy on the COSMIC mutation signature webpage is we give information about how different signatures have been validated. Have they been replicated? Have they been seen in different sequencing methods, different variant colors, and also in multiple sequencing centers? So COSMIC can help you answer the question of when a mutational signature is ready to shift from discovery to clinical use. So here, for example, on screen, I'm showing a slide for platinum chemotherapy-induced mutational signatures. I can also go ahead and just share my screen to show that it's possible to pull this information up, where if I go and browse the various single base substitution signatures, this is where I'm able to see quite a variety of potential signatures. You can also download these signatures as well. But here, if I go ahead and pull up this proposed platinum chemotherapy treatment signature, this is where I can see details about the signature itself, and also this incredibly useful chart where I'm able to see this is something that has been found. The etiology has been confirmed. So in this case, it is platinum chemotherapy induced and confirmed by experimentation. That experimentation has been published. And again, just the level of reproduction is clearly indicated. And this can be especially useful when you're dealing with an emerging signature. So for example, this thiopurine chemotherapy signature, it's one where the proposed etiology has been experimentally confirmed. In this case, it's been published in Blood. However, the technical validation hasn't been completed yet. So it has been found with orthogonal methods, but it hasn't been replicated. And the sequencing methods that one could possibly use in the clinic to diagnose this haven't been shown to be through different methods. We haven't received evidence that these mutations are detectable with different pieces of software. And we also haven't run this through multiple sequencing centers to account for instrument bias. So overall, COSMIC can help you stay current with that cutting edge and also assess when do these signatures shift over into being something beyond just for research. When can you use them to potentially take actionable steps? So just to summarize overall what Steve and I have presented today, I hope we've given you a great introduction to all of the data that COSMIC and QIAGEN have to offer. Hopefully, I've shown you some of the benefits to taking advantage of the COSMIC downloads, whether you're an academic or a clinical researcher. Steve showed many of the excellent curation details present in the Cancer Mutation Census and the actionability details. So those are all things that can be really valuable just in the amount of time saved because someone has already taken the time to find those details and carefully summarize them. And then finally, that same level of care is being applied here for the cancer mutation signatures, which hopefully are something that will emerge over time and gain more utility. So with that, I want to go ahead and stop for a final question break. And again, thank you all for the time. I'm going to go ahead and just put our contact information up on screen while we receive those last questions.
All right, fantastic. Thank you so much, Dr. Nilsson. And thank you, Dr. Jupe, for your presentation as well. Let's see, we have a lot of questions. We're definitely running short on time, but we will go ahead and address a few of them. And then again, if we miss any, we will email those answers out after the conclusion of the webinar. So first question is, the first few actually are for you, Dr. Jupe. How often is actionability and cancer mutation census information updated?
The CMC is updated at every COSMIC full release, which is 3 times a year. And the actionability data is updated every time there's an actionability release, which currently is 4 times a year.
All right, next question. How are the contradictory consequences for specific mutations published in literature handled in COSMIC curation?
Okay, that's a good question. I think at the moment in the COSMIC core database, if a variant is associated with a sample, then it's recorded in COSMIC. There's no actual sort of association of, do we believe that variant is a driver, is it a bystander? It's just that simply recording the fact that that variant was discovered within that sample and therefore recorded so that it becomes part of the accumulated frequency. If you're assessing the significance of a particular variant, that's when you'd need to go and look at CMC, where CMC will take variant frequency into account when it's assessing the tiering, but other measures as well, things like is there active selective pressure from evolutionary pressure on the particular variants? Are there other does the ClinVar suggest that it's a likely pathogenic variant or not? And several other measures as well, all combined to give you an overall indication of whether this is likely to be a driving mutation or not.
All right, thank you. Next question, are mutational signatures included in cosmic actionability?
No, they're not actually part of the actionability data set. They're something separate.
Okay, and the next one says, I may have missed a couple of slides. Do you hand pick the actionability scores?
Well, they're calculated from the clinical trial information that we've been able to find.
Okay, let's see. Next, where can I find DMD information in Cosmic?
Oh, the DMD, that stands for drug mutation disease, and that's within the actionability data downloads. It's really just an internal identifier that's used as part of the annotation process.
All right. And final question that we're going to take for right now is, are there any differences between commercial and non-commercial Cosmic data downloads?
No, if you have the license, then it doesn't matter which kind of access you have, you get the same data.
And if you find that you do have issues accessing any particular feature, let us know so we can go ahead and fix that.
All right. And that actually concludes our session. So thank you again, Drs. Nilsson and Jupe, for your time today. Thank you as well to the audience for joining and for your active participation. We really appreciate it. We hope it was mutually beneficial. Again, any questions that we didn't get to, we will address via email, the email that you provided at registration. Obviously, our contact information for both QIAGEN and Cosmic is also up on the screen for anyone who might want to reach out with additional questions. Again, you will be notified via email when the recording of the presentation is available for replay. We highly encourage you to share that with any of your colleagues or anyone else who may have missed it and would find it interesting or useful. Until next time, stay safe and be well, everyone. Goodbye.
Thank you. Goodbye.
Bye-bye.